again with dye added to showing the flow, the animal is going to react very strongly, this time in only 7.5 times time lapse, because of course with slugs you always work with time lapse, and to show you what the slug does in response. So here I've added the sea star, I've put some dye on it, you can see the flow, and the slug immediately reacts, turns away downstream, and sprints away for a sea slug. It is crawling as fast as it possibly can. Now, of course, I've been messing around with a sea star and dye, so it could be just me. So since we are now manipulating things, we did a proper control treatment, and this time I'm using a dive glove instead of a sea star with dye, and the flow did not cooperate very nicely, trying to find where upstream and downstream are. But you can see that sea slugs do not care about divers messing around with things other than sea stars around them. So like I said, we did hundreds of hours of this, analyzed the directions the animals crawl, and it became very clear. The animals are using what appears to be Odour-Gated rheotaxis in their natural habitat to find prey, to find mates, and they reverse it. They crawl downstream rather than upstream to avoid predators. So to really conclusively prove that they're using this strategy, Odour-Gated rheotaxis, is actually quite difficult. And I'm just going to do a little bit of an explanation of how we've done some of the experiments to target this. I'm not necessarily going to get into the full details. You can ask me about it at the end if you want to know it. So one of the things we can do is play around with the animal's sense organs to help understand whether or not they're using Odour-Gated rheotaxis, because unlike most navigational strategies that work, I shouldn't say most, unlike chemotaxis, Odour-Gated rheotaxis can be done with just one sense organ. Chemotaxis, detecting odor gradients, almost certainly involves an animal detecting what the odor concentration is in one location and detecting it in another. And that means they either have to move their body in a way to detect two different locations, or more usually have two sense organs. Odour-Gated rheotaxis, on the other hand, only needs one sense organ, because you don't need to know how much concentration of odor there is, you just need to know it's present. So we look at the face of the slugs. They've got these two obvious tentacles. They're called Rhinophores. And they got this great big mustache called the oral veil. And we did some simple surgeries after anesthesia with these animals and showed that those tentacles, the Rhinophores, are necessary for this navigation behavior. We've moved the animals into the lab, into a flow tank. We've got flow from top to bottom here. And here I'm going to show you an intact animal encountering an odor plume and crawling straight upstream towards it. We're piping in the C-pen odor right here. If we surgically remove the Rhinophores, the animals show absolutely no response to the odors. So this tells us the Rhinophores are necessary for navigation. Here's some summary diagrams of tracks animals took if we released seawater, prey odor, or predator odor. And you can see if we put seawater into our flow tank with flow coming down this way, the animals start down here. This is the track of one animal. There's in fact 10 to 12 animals in each of these diagrams. And you can see seawater, the animals go all over the place. If you put prey odor in there, they really head towards it. And if you put predator odor in, they avoid it. So this is what intact animals do. So my student Greg then did a whole bunch of surgeries. He removed Rhinophores, or in fact, what he did was he cut the nerves that led to the Rhinophores. He did some sham surgeries. He removed the innervation to the oral veil. And in fact, he did remove the innervation to one of the Rhinophores and one of the oral veil, leaving just one intact Rhinophore. And this is a complicated diagram. The key is stand back or lean back and notice that all of these here look roughly the same. Every one of these 6 squares look roughly the same. Seawater, Prey, again, they all look roughly the same. They're able to find the odor source. And in predator odor, they avoid the odor source, regardless of what Greg did. So that means the animals can navigate with just a single sense organ. And that's what really gives the strongest evidence to date that these animals are using this Odour-Gated rheotaxis, because this single rhinophore, or alternately this one, they can do it with either, but they don't need both of them, can detect whether or not odors are present. They don't need to know what the concentration is. They just need to know that it's present, and then they can detect which direction the flow comes in one of these. And that tells them which direction to go when they detect the odor, Odour-Gated rheotaxis. Okay. So, sea slugs use Odour-Gated rheotaxis. More recently, we've been doing some work with snails to look at how their navigation behavior can be augmented by other senses. These are pond snails. They live in interesting environments. They live in swamps, ditches, and streams in their natural habitat. They eat aquatic plants, which are abundant. In the lab, we can grow them with romaine lettuce. And they tend to live in these low flow environments. But in fact, they live in a wide variety, all the way from essentially entirely stagnant water to some reasonable flowing environments that might even get turbulent. And we've done a whole bunch of experiments that show that the animals can navigate in no flow, in laminar flow, slow flow environments, and higher flow turbulent environments. This is all done in the lab. So if we stick them in a circular dish with no flow at all, and we stick an odor source in the middle, they go and find it. If we put them in a flow tank and create a laminar odor plume, they go and find the odor source. And if we speed the flow up so that it goes turbulent, they can still find the odor source. And so what we think is going on is they're actually able to switch between chemotaxis and Odour-Gated rheotaxis, depending on the flow conditions. Quite what would they do in laminar odor, we don't yet know, because either would work there. So I'm not going to go into the details of the experiments of that. My MSc student, Reed Al-Ansari, has spent an entire master's figuring all that out, and she's soon going to be defending. What we did in addition to that is explore the effect light might have on these behaviors. So I'm switching to a different way of visualizing how the animals navigated here. It's a easy way to show what lots of animals did, because when you deal with slugs, things can go very slowly. So I'm showing you a video here, sped up, of a snail completing some navigation over 30 minutes. And trying to keep this in your head about exactly what that animal did can be quite difficult because it takes some time. We can capture exactly what it did in this heat map. It spent a little time over here, then it sped up and spent very little time along this track, and then it really hung around the middle here because there was something it was very interested in there, a protein pellet that they like to eat. And so this is one piece of evidence that they can do chemotaxis to find food in still no flow. We can then take this trial and superimpose it over top of all the other trials that we ran this experiment with. What we can see is that the animals all started up here and spent a lot of time. These hotter colors are telling us where they spent more time. So they were really quite attracted to that protein pellet placed inside the cage in the middle there. So these heat maps show us what the animals do in the different treatments of our experiments. And so that's what this shows. They can find odor sources in still water. I already told you this. This is what Arij did for her master's. So if we have no odor source in the middle, the animals spend most of their time on the edges, having started down here. If we put lettuce in the cage, they spend more time around the cage. And if we put that protein pellet in there, they spend even more time. And remember, this is a composite diagram showing the activity of about 18 animals, 18 other animals, and 18 animals. What's interesting is if we compare that data, it's the same data right here. Okay, no odor source. Sorry, not the same data. This is a second experiment by Hannah where she's replicated Arij's work, showing with no odor source or the cage, and then protein food, we get very strong attraction, minor attraction, and no attraction. Then if we do it all again in the dark, this time lit with infrared light so our cameras can still capture them, the animals show no responses at all. And so this shows us that light is playing some form of role in the navigation behavior of these aquatic animals also. We don't know whether it's simply a motivational cue. Are they not interested in navigating towards food when it's dark? Or is it got something to do with initiating the navigation? They don't start moving towards the center where they can start picking up that diffusing chemical gradient of odor from the protein pellet. Okay, they need to see the cage first, move towards it, then detect the chemical. We're not sure. What we do know is that if we repeat the experiments in a flow tank, they will still respond in the dark. So this interaction with photoreception, detecting light, whether it's seeing the cage or just the overall detection of ambient light to motivate them, we're not sure which, only applies when the animals are in a no-flow environment. We're not quite sure what that one means. Would welcome thoughts on it at the end. The animals almost certainly are using gravity as well at the same time. Lymnaea is a snail, but it is an air breather. It is an aquatic snail that evolved from terrestrial ancestors. And so these guys all have a lung that they move up to the surface of the water to fill periodically. So that's what these animals are doing. They're going through the respiratory behaviors. Oops, again. Going through the respiratory behaviors that these animals use periodically, moving up to the surface, taking a breath, and coming back down. And that movement up to the surface almost certainly involves a response to the direction of gravity. And so that means they have to have a gravireceptors, receptors that detect the direction of gravity. And what other people have shown is that if you put them in the presence of a predator odor, either from fish or crayfish, which eats the snails, they'll increase the frequency of the aerial respiration, so they'll go up and take breaths more often. And then they'll also congregate at the surface, either just below or even just above the water surface. Both of these are thought to be avoidance tactics. So with this in mind, and what we found with the sea slug, we wondered whether the animals might have additional responses beyond this negative geotaxis, moving against the direction of gravity. Gravity goes down, so they move up. That's negative geotaxis when they detect odors. But maybe they also avoid odors from predators and flow the way the slugs do. So Carmen Uchiferi spent a summer testing this and found no such response whatsoever. So in no flow conditions, we've got an empty crayfish cage here, and then we've got a crayfish inside a cage here. And you can see the snails essentially did not care. They went in all over the bowl and are all over the cage in both cases. And then in our flow tank, where we have the odor source here and the flow moving left to right, again, the difference between without and with a crayfish present generating odors versus no odors, no difference in the behavior of the animals. So the Lymnaea does not seem to avoid predators in the sense of trying to stay away downstream from them. They do seem to only move upwards. We did find some evidence for that in our measurements, just as others had shown before. So, putting it all together, it appears that these simple snails are doing chemotaxis sometimes, probably Odour-Gated rheotaxis, or some other complicated version of chemotaxis that I'm not going to get into right now. So they either follow chemical gradients or they follow a flow when they detect odors, depending on the flow conditions they encounter. They'll also kick in Odour-Gated geotaxis. They detect an odor of a predator. They'll move upwards. There are influences of vision on these behaviors, but those are not straightforward. We've got to unpack those some more. And undoubtedly, there's some tactile influences also, because the process of finding a wall or plant stalk and crawling up it almost certainly involves some tactile sensing also. So lots more